What is GeneNetwork? GeneNetwork is a web service that's used to analyze the relationship between differences in genotype, genetic differences, SNPs, indels, copy number variants, and differences in phenotype. When you open GeneNetwork, this is the screen that you should initially see, the select and search screen. It allows you to select among several species, human, mouse, rat, and 2 species of plant. We have the most data for mouse, so I'm going to leave it in the default position, mouse. There are several families or groups of mice that have been studied and for which we have data in GeneNetwork. We have the most data for BXDs. These are other families. Some of these are F2 intercrosses, others are panels of recombinant inbred strains. For each one of these families, we have some types of phenotypes, classic phenotypes, genotypes, and often RNA expression data sets. The initial default is to this hippocampus RNA data set, but we can change that easily to classic phenotypes. To search for a term, one can either enter a single term or a group of terms into the Get Any field or in the Combine field. So for example, if I wanted data for brain weight, this would be the best place to enter the search terms. It will then combine these and search for any data on brain weight that happens to be available for the BXD strains. I can select several of these records and put them into a collection, or I can simply click on one of these records and get the data itself. So these are data for brain weight from the BXDs and the parents. Here's one of the F1 hybrids that's actually used to make the BXD strains, the maternal strain, the paternal strain. You can see a very large difference in brain weight between the 2 parents. And then the progeny strains, some just like one of the parents and others more like the other parent. It's not convenient to look at the data in this particular format, but it is possible simply to click on the basic statistics button, and this will produce a summary of the data. We have data for 88 strains, a box plot, the range. Here's the minimum brain weight, the maximum brain weight. The box plot shows you that one strain is an outlier, a high outlier. These are what the data look like in order of strain name, starting with the F1s and going through to BXD 103. Next panel down, same data but organized from low to high, where BXD 5 has high expression, BXD 27 has low expression. These are standard error bars for each of the strains. This is a QQ plot that just, if it's a straight line, indicates that it's more or less normal distribution, which these data are more or less normally distributed. With data of these type, you can ask quite a few questions. You can ask, what is the genetic, can you map the genetic sources of this difference in brain weight? And that's done using this interval mapping tool or marker regression tool. Or you can ask, what does brain weight covary with? What other traits covary with brain weight? And I'm going to click on the trait correlation button to compute the correlation between the data we just looked at down here and any one of about 2,000 other phenotypes that have been measured that are in this published phenotypes database. But one could also select any of the RNA expression data types or even the BXD genotypes for that matter. And the correlations, you can either select Pearson product moment correlations, rank order, Spearman correlations, and several other types of correlations. But let me just leave it at the default. Click on that button, and now it's computing the correlations between brain weight and other traits. And of course, it comes up with a correlation between brain weight and other measurements of central nervous system weight, You can see these right here, very high correlations. This is from a single study, and that's why the correlations are so remarkable. But as we scroll down here, we find, for example, here's a correlation with a study in 1999, a study going back to 1992. For example, here's a correlation with work done by John Belknap and colleagues in 1992. I can actually click on this correlation, and it shows me how today's data covaried with data a long time ago. And you can see it's a very nice correlation with BXD5, the brainiest strain consistently, and BXD27, one of the smaller brain strains. So I've shown you the basic statistics button, the trait correlation button, and now we can look at the mapping button. The mapping button, or interval mapping, will determine to what extent variation brain weight is associated with genetic markers along the genome of the BXD family. So just click on that, and it is now computing the interval maps and performing 1,000 permutations that are used to determine the significance of the results. So this is what the mapping result looks like. There's some parameters you can tweak here. On the x-axis are the mouse chromosomes, chromosome 1 through the X chromosome, and on the y-axis is the likelihood ratio statistic that there is a sequence variant or sequence variance on a particular chromosome that modulate brain weight. And in this data set you can see that there's a hot spot on chromosome 19 that's likely to modulate brain weight. If you actually click on the number 19, we can focus on that particular chromosome. This is what a quantitative trait locus map looks like for chromosome 19 and brain weight. Up here, these little colored blobs are genes, or they're scattered. You can see there's a gene-rich region right here, a very gene-sparse region over at about 50 megabases. Our peak linkage is right about here at 10 megabases on chromosome 19. I can click on this pink area to zoom in on that. And below this, by the way, are all the genes on chromosome 19. In this case, about 1,200 genes that are known to map to 19. This is the blow-up of chromosome 19, and we can now see that our peak is actually in the 10 to 14 and beyond area. So that's how we generate a QTL map for a trait such as brain weight. Let's have a look at how we would, let's look at the correlation between brain weight and gene expression in the brain. So again, we're looking at our brain weight data, and we're going to look at the correlation between brain weight differences and the expression measured by the INEA consortium using the Affymetrix M430 array and the RMA transform. I click here, and now it's computing the correlation between brain weight and about 45,000 measurements of gene expression in the brain. Results. So these are genes that covary with brain weight weekly, 0.6. I guess you could argue that that's not that weak. 42 cases in common between the two data sets, the brain weight data set on the one hand and the INEA expression data. We can click on any one of these, so let's click on this one to look at the structure of that correlation. And you can see a lot of it is driven by this one outlier. We can now begin to assemble networks. So let me click on several of these. Click on the top 10 covariants. I'm going to then add them to my shopping cart. And you can see I had previously added data on brain weight to the shopping cart. So now we have a collection or shopping cart with two types of traits. It has the classic phenotype data as well as gene expression data. And I can begin to ask some questions about whole networks of traits. If I click on the matrix icon here, it'll generate a correlation matrix of all of these traits. I can add long labels so that I know what they are. And so if I'm interested in the correlation between obscurin and brain weight, I would go down here. This is that particular correlation. And I can click on any one of the items in this. Spearman rank order correlations are over on this side, and Pearson product moment correlations on this side. So that gives you an introduction into whole sets of traits. So I think that'll give you a feel for what data types are in GeneNetwork and how you can begin to play with the data. In the next video, I'll show you more sophisticated analysis that can be done with these data sets